I'm from NASA Goddard Space Flight Center, and I just want to say right off the bat that I'm actually an organic geochemist and astrobiologist. This is my first foray into thinking about the moon, but I'm delighted to work with many lunar scientists and contamination control engineers that you see listed here on this slide. And I'm going to be talking about something potentially very different. Now for something completely different. Next slide, please. Yes, I'm going to be talking about potential habitable niches where microbes could potentially survive and maybe even grow on the moon. We generally think of the moon as a fairly harsh landscape, not particularly amenable to life, even microbial life, but actually there are many organisms that have adaptations that would confer a certain amount of survivability in those conditions. And we know this from experiments that we've done with these organisms on places like the International Space Station, where we found that not only can they survive um, uh, space travel, but they can actually survive on the exterior of the spacecraft as well. We also know there's a lot of organisms that can survive bioburden reduction protocols, planetary protection protocols that we use to clean spacecraft. And interestingly, many of these organisms aren't the rare extremophiles you might think they would be. They are what we would call cosmopolitan microbes. That doesn't mean they're sitting around with martini glasses. It just means that they're ubiquitous. They're everywhere. So even though they're not the rare ones that we think of as having these special properties that would give them survivability advantage in space, they're not the ones that you find in Yellowstone or Atacama or Antarctica. They're replete. They're probably on you right now. And that means that there's many of them and they're difficult to kill. And so we have to think about what this means for lunar exploration. Next slide, please. Here, I'm just showing a list of some of these taxa that I'm talking about that have some of these special survivability skills. This includes both bacteria and fungi, including yeast, which of course we all know is ubiquitous and found in many, many locations. This list includes organisms that are bacteria and fungi that have been demonstrated to be able to survive in open space. We also see some here that we know survive the microbial bioburden reduction protocols, as well as taxa that we know have this capability. They haven't been experimented on in space, and we haven't necessarily done studies to understand their survivability when it comes to planetary protection protocols, but we know that they have many of these same advantages. And you'll see here also the ubiquitous taxa that I'm talking about that are really everywhere. They're found on medical equipment, they're opportunists, they're common contaminants, and they have diverse metabolisms and some really clever ways of surviving difficult conditions. Next slide, please. Before we talk too much more about these particular taxa and where they might be found on the moon, I wanted to discuss what I mean when I'm talking about survival versus growth when we're talking about microbes in space. Could I have the next slide? And this is really getting to this point about a scale of lifiness, where we're going from something that's very non-living, like a rock, up into the scale of a dorm room fridge. And I used the least offensive picture of a potential dorm room fridge that I could find. Next slide, please. And for many of these organisms that we're thinking about, we would be thinking about them as being very much on the lower end of this scale of lifiness. We're thinking about viruses and spores. Viruses, of course, don't have the potential to reproduce on their own without hosts, so they're not really something that we think of as being a contaminant in that way, because they would need an ecology. And spores. Spores are something you probably hear about a lot when we're talking about planetary protection. And next slide, please. Just so everyone knows, since I know not everyone here is a biologist, a spore isn't necessarily a cell. A spore is an encapsulated part of the cell that is basically a lifeboat. So when times get really hard, certain cells, not all cells, have the ability to create this encapsulated, very hardy, mummified version of itself. And these spores can last in really harsh conditions for very, very long periods of time. Some suggest up to hundreds of years. But then when times get better and there's access to energy and nutrients and the physical conditions are better, next slide, please, these spores can generate into cells, and that's when we have growth. So this is where we think of survival versus growth. Spores are surviving, cells are growing. So when we think about lunar exploration, we can think about spores as being a different kind of contamination problem than growth. Spores represent a certain amount of false positives if you're thinking about certain organic analyses, things like that, especially since they're very hardy on certain landscapes. But growth is another condition that would need nutrients, energy, better physical conditions, and even in most cases, many cells so that you could develop an ecology and grow up to be, a dorm room fridge. So I just thought I'd make sure that's clear since not everyone here is a biologist. Next slide, please. So we usually think of the moon and the surface of the moon and the physical conditions there as doing a lot of the killing for us when we're trying to think about preventing organisms from surviving or growing. There are lots of features about the surface of the moon, as you all know, that would prevent growth. So we have huge temperature extremes, UV radiation, energetic particle radiation, and even hard vacuum. And this is a big pressure for most organisms to survive. And we have modeling work that supports this. You're probably all very familiar with this work. And so we think of all of these conditions as something that are going to prevent growth and even spore states. However, a lot of this modeling work hasn't considered topography, and so we're using data that comes to us from LRO and LEN and other remote sensing sources to help us understand the effects of this topography and the angle of the sun and the moon, to think about shadowed regions and places that might confer a certain niche where survivability is possible. Next slide, please. So here is just, here's a demonstration of what that would look like. If you think about the sun's low angle and the combination of topography, this means that there would be relatively colder year-round surface temperatures in these shaded regions that are related to this topography change. This might be considered one of those micro PSRs that Noah talked about at the beginning of the day. And you can see examples of this here from some of the imagery that's taken with the U2 rover, as well as Apollo 17 imagery. And that diagram at the top is trying to show those partially shaded regions as well as that completely shadowed region at the bottom, which could provide a fairly well protected niche for an organism. So this is a case where the low elevation of the sun combined with topographic relief is promoting survivability through a radiation refuge. Next slide, please. So here is some of the data that my colleagues and I have been looking at, where we are looking at the polar max temperatures, trying to find locations near the Artemis landing sites, which are the boxes on these images, to think about where there may be temperatures that would actually be clement enough and not too hot for microbes. Most of the conditions that we think of as being harmful to microbes on the surface of the moon would actually be the higher temperatures, not the lower temperatures. Many spore states were not actually sure what their lowest temperature range that they're survivable at is. And we routinely store them at -80°C. We're also mapping out polar water equivalent hydrogen to think about potential evidence of surface and subsurface water that could confer survivability. And then those bottom two panels are showing us the surface roughness and sun visibility fraction as a way to think about protection from UV radiation and energetic particles, which could provide niche space for these potential microorganisms. And so you can see that in all of these cases, some of these conditions that would actually confer survivability overlap with the potential Artemis landing sites. Next slide, please. This slide is where we have taken thresholds for a certain kind of organism, a cosmopolitan, common contaminant on you right now, Bacillus subtilis. It's a spore former. It's one of the organisms that we're looking at that does form spores, and looking at places that will never exceed 30C and a sun visibility maximum of 10%, and thinking of that as our threshold of survivability for these organisms and how that overlaps with the Artemis landing sites. So this low sun visibility percentage was really chosen as a way of thinking about UV flux and solar radiation, and that's not necessarily based on experimental data, whereas the temperature limits are, although there is some evidence to suggest that these could actually survive up to 50C. So next slide, please. Finally, I just wanted to summarize with a few points that I'm hoping you take away from this, is that all of these areas of interest could have potentially habitable niches or micro niches in some of these micro PSRs. And so this becomes something to think about if spore survival or contamination would be a problem for a study that you want to do at that site. For example, if you're going to do any sort of organic analysis. We could also think of these habitable niches as being a key metric for assessing and tracking contamination during exploration. And I wanted to let everyone know that we're taking all of these surface properties and all of these survival thresholds from the organisms of interest, and we're putting together a statistically framed analysis of all of these sites, and hope to get a paper out to you soon. Thanks.